Welcome to this comprehensive lecture on the fundamentals of biomolecules. Today, we are going to explore the fascinating world of carbohydrates, which are the essential molecules that fuel and build life on Earth. We will uncover their intricate chemical structure, understand their immense biological importance, and learn the mathematical rules that govern their shapes and forms. Let us dive right in. To start, imagine you are building a house in the cold winter. You need sturdy bricks to construct the walls, and you need dry firewood to burn inside to stay warm. In the biological world, carbohydrates serve exactly these two vital purposes. They are the quick-burning firewood that gives your body the immediate energy to run, think, and breathe every single day. At the same time, in plants and insects, they act like sturdy bricks, building strong stems, leaves, and protective shells. They are nature's ultimate multi-purpose tool. Now that we understand their purpose, let us look at how they are built. In biochemistry, carbohydrates are formally defined as polyhydroxyaldehydes or polyhydroxyketones. While that sounds intimidating, it simply means these molecules have a carbon backbone decorated with many hydroxyl groups, which are oxygen-hydrogen pairs. They also feature a carbonyl group, where a carbon atom is double bonded to an oxygen atom. Because they are essentially carbon chains heavily decorated with water-like attachments, they earned the name carbohydrates, or hydrated carbons. This specific arrangement of atoms leads us to an important chemical classification. Depending on exactly where that double-bonded oxygen, known as the carbonyl group, is located, the sugar gets a completely different name. If the double-bonded oxygen is positioned at the very end of the carbon chain, we call the sugar an aldose. However, if it is located somewhere in the middle of the carbon chain, we call it a ketose. Ultimately, these molecules form long chains, or even fold into rings, creating a massive variety of structures. Before we move on to the math, let us see why these structures matter in the real world. When you eat an apple, your body breaks down complex carbohydrates into simple sugars like glucose, which powers your brain and muscles. Without them, our cells would starve. In plants, a tough carbohydrate called cellulose allows them to stand upright. Furthermore, carbohydrates attach to the surface of your cells, acting like molecular ID cards. This helps your immune system instantly recognize whether a cell is a friend or an invading foe. Understanding their biological role brings us to their mathematical representation. To understand carbohydrates chemically, we first look at their general empirical formula. The formula is CnH2ON. Conceptually, this tells us that for every single carbon atom in the backbone, there is exactly one water molecule attached. The symbol N represents the total number of carbon atoms. If the value of N increases, the molecule physically becomes larger and heavier, transitioning from a simple sugar to a massive polymer. This simple ratio perfectly captures why they are historically called hydrated carbons. Now that we have our formula, let us see this with a concrete example. Suppose we want to find the exact chemical formula for a simple sugar that has exactly 6 carbon atoms, such as glucose. We are given that the variable N equals 6. We substitute the number 6 into our empirical formula. By multiplying the 2 hydrogen atoms and 1 oxygen atom inside the parentheses by 6, we find the exact atomic inventory. The final answer is C6H12O6. This represents the exact physical composition of a single glucose molecule. This chemical formula we just used leads us to another crucial concept, the three-dimensional shape of the molecule. In nature, a single chemical formula can arrange itself into many different 3D shapes, which are known as stereoisomers. The 3D shape is absolutely critical because it determines how a sugar tastes, how it is digested, and how it functions in the body. The number of possible shapes depends entirely on chiral centers, which are specific carbon atoms attached to 4 completely different chemical groups. To find out exactly how many 3D shapes are possible, we use a specific mathematical equation. The formula is n equals 2 to the power of nc. Here, the symbol n represents the total maximum number of possible stereoisomers. The variable nc represents the number of chiral centers in the molecule. The base 2 represents the 2 possible spatial orientations, which are left or right, at each chiral center. As the number of chiral centers increases, the number of possible shapes grow exponentially, showing how nature creates massive variety. Let us apply this formula to our 6-carbon aldohexose, glucose. We need to find the number of chiral centers. Carbon 1 is part of a double bond, and carbon 6 is attached to 2 identical hydrogen atoms, so neither is chiral. However, carbons 2, 3, 4, and 5 are each attached to 4 different groups. This gives us exactly 4 chiral centers. We substitute 4 into our equation, calculating 2 multiplied by itself 4 times. The final answer is 16. This means glucose is just one of 16 completely different 3D shapes possible for this formula. Now that we understand single sugar molecules, let us look at what happens when they join together. When simple sugars link up to form a larger chain, they undergo a chemical reaction called dehydration synthesis. This term literally means building by losing water. Every time a chemical bond is formed between 2 sugar molecules, one molecule of water is extracted and removed. Because water is lost during this bonding process, complex carbohydrates deviate slightly from the basic empirical formula we learned earlier. Let us calculate the exact formula when 3 glucose molecules fuse into a trisaccharide. First, we multiply the glucose formula by 3, giving us a starting inventory of 18 carbons, 36 hydrogens, and 18 oxygens. Next, because linking 3 molecules in a row requires exactly 2 connecting bonds, we must remove 2 molecules of water. This means we lose 4 hydrogens and 2 oxygens. We subtract these lost atoms from our initial inventory. The final answer is C18H32O16, proving why complex sugars become more compact. To visualize everything we have discussed, look at this classification diagram. At the top, you can see carbohydrates branching into monosaccharides, disaccharides, and polysaccharides, depending on how many sugar units are linked together. Below that, we see the actual chemical structure of glucose. Notice how it can exist as a straight vertical chain on the left, but it can also fold into a stable hexagonal ring structure on the right. This ring formation is how sugars typically exist in your bloodstream and in nature. We have covered a lot of ground today. Let us review the key takeaways. First, carbohydrates are the fundamental fuel and structural framework for all living organisms. Chemically, they are polyhydroxyaldehydes or ketones, heavily decorated with hydroxyl groups. While simple sugars follow a strict hydrated carbon formula, their 3D shapes vary wildly due to chiral centers. Finally, when these sugars link into massive chains, they lose water through dehydration synthesis. You now have a solid understanding of carbohydrate structure and importance.